Hey, well, first I'd like to thank the organizers for the opportunity to present today, the SENS Research Foundation, and Jerry, who was helpful with all the logistics. Secondly, I'd like to say that I am a cell biologist, immunologist, in the same sense of the word that a woodpecker is a carpenter. So save your hashtag questions for some of the distinguished speakers that follow or have preceded me. I'd like to tell you a little bit about, let's see if I can get this working. Yeah, I'd like to introduce WuXi App Tec, and I know WuXi App Tec is not a household name, particularly in the space of advanced therapies, so I want to spend a little bit of time telling you about what we're doing. And then my challenge is to cover all of the challenges of manufacturing these innovative products. And those include the facilities, the chain of identity, scheduling and logistics, the quality control paradigms, the process development that is required to get to a commercializable process, and also the manufacturing partnerships that are going to be critical going forward given the complexity of these products. And I'm going to do that through the lens of a autologous CAR-T immunotherapy that's being manufactured in our facilities. WuXi was founded in 2000, has grown very rapidly to include 12,000 employees and more than 5 million square feet of laboratory space. And it's speckled all around the globe, including Reykjavik, Iceland, where the Decode Genetics and the genomic programs are residing. Our sites in Philadelphia are the center for excellence in biomanufacturing and biologics testing, and we participate in other activities such as supporting combination devices and medical devices with our St. Paul facility and our Atlanta facilities that are providing sterility assurance services. WuXi is committed in a large way to cell and gene therapy, and we're supporting programs that represent both large multinational pharma companies as well as biotechs and small innovative companies, about 2,000 and spanning 30 different countries. In terms of what we're doing in Philadelphia, we've set up an integrated service within the Philadelphia Navy Yard to provide for both manufacturing and quality control testing and analysis of cell and gene therapies. We have initially utilized the first facility in the Navy Yard, but have now brought on 2 additional facilities that are earmarked for the production of these very complex products on a commercial basis. And so these are large-scale facilities. Our services span the development spectrum from discovery to preclinical and hopefully clinical and commercial. And we're supporting phase 3 manufacture of advanced therapy products right now for use in the United States, Korea, and Europe. I mentioned WuXi's commitment to this very exciting area. Our initial investment in 2004 was to build structure for the production of autologous and allogeneic cell therapies and the associated biosafety and quality control components that were required for those products. We recognized as our clients' products advanced that these facilities were going to be inadequate, and so we, in 2014, started construction of a 45,000 square foot facility for allogeneic and autologous products that do not include the use of viral vectors. And as excitement in the CAR-T space was expanding, we were committed by our management to enter that field, and I had the headache of making that happen. We concurrently broke ground on a new facility, 150,000 square feet, that is earmarked for commercial manufacturing of CAR-T products and viral vector manufacturing. These facilities, and FDA has very specific requirements. These are products which require aseptic processing. And we must meet those aseptic processing standards in order to release products and to pass a pre-approval inspection by FDA. It turns out that the US has been more lenient than the EU in terms of their requirements for clean rooms. And so we took the decision to build all of the new facilities to meet the EU standard. And this requires a greater expense, greater stringency, but it is, in our minds, the most appropriate way to move forward. Cell therapy covers a very broad field, and you've heard lots of different terminology. I'm going to be talking about CAR T cells, which we describe as gene-mediated cell therapies. And we're using gene-mediated cell therapies to also describe products where there is gene editing involved in cellular expansion. As you heard from Jean this morning, and certainly from others that will follow, there is complexity associated with all of these different kinds of cellular therapies. The requirements for control of a manufacturing process that's driving cells to differentiate to pancreatic islet cells is very challenging as well, and it has different ramifications in terms of facilities, staffing, and scale-up. From a manufacturing perspective, allogeneic products have a commercial advantage in the context that you can make lots of patient doses from well-characterized banks of cells, and you can deliver more patient doses to the patient. The autologous products, though, increasingly have yielded results that are very stunning in the clinic. And just, I think, in an article in the New York Times yesterday, Steve Rosenberg said we are going to get to the point where we're going to be excessively personalizing these therapies. So we believe that the autologous products are going to be on the horizon for a good while. The autologous products have a whole set of challenges. It includes limited amounts of material that one can test, source variability, and often the fact that these cells have to get back to the patient very quickly, in which case we need to use rapid test methods and to quickly review and release product under GMP. This is a different cartoon, and I apologize. You're going to see some similar graphics across our slides, but this maps out a gene-mediated cell therapy, whether it's an autologous or allogeneic cell therapy. Goes through a very similar process in terms of collection of cells, selection, ultimately the transduction with viral vector, and then harvest of those cells. We're also supporting cells which start from the source cell bank, are genetically engineered, and then are used for expansion and harvest and given to the patients. When we looked at bringing in CAR T manufacturing into our manufacturing environment, there were two issues that stood out as requiring the most attention. One was the ability to appropriately handle the retroviral or lentiviral vectors that were being used as part of the transduction. And the second was ensuring adequate segregation and chain of identity for individual patient products in that same environment. We took the approach of establishing multi-workstation suites, aseptic processing suites, and having very careful mapping of activities within those suites. And this is an example. It shows you 4 workstations within an aseptic processing environment, and mapping of workflow across the different workstations to shared equipment. And needless to say, much of the equipment is expensive, and so where possible, we want to use shared equipment or shared workstation space in order to get the highest throughput through the facility. And this requires a good deal of discipline, and it requires training so that our operators understand the temporal segregation requirements and the non-overlap in important unit operations. FDA expects that we as a manufacturer will have adequate procedures to ensure that cells collected from a donor go back to that same donor after they've been processed. And it sounds simple, but the reality is there are lots of places along the way that require a good deal of attention and special processes in order to accomplish that. We've broken it up into a program for each individual product that we support and looked at various quadrants such as the way that we segregate activities, the way that we label materials, samples, derivative samples, the way that we monitor the process and verify that the chain of identity is being maintained, and the storage and transportation of those materials back to the individual patient. That necessarily requires us to be in communication and to have coordinated planning and project management with our clients and their clinical team. So it is a very challenging task, but it is one that has successfully been implemented. And for each new product that comes into our facility, we establish a cross-functional team that evaluates the workspace, the equipment requirements, does essentially a gap analysis, and then looks at filling those gaps with individual processes or procedures that become part of the overall operator training. And we train not just the operators, but the logistics staff, the quality control staff, the analysts that are receiving these samples, so that there is a comprehensive appreciation for maintenance of the chain of identity. And this documentation ultimately ends up in regulatory documents supporting the product. I'm not going to describe the T-cell process. I'm going to talk a little bit about the mechanics of how we carry this out in the laboratory. And I think it's important to recognize that in this processing, there are variable steps and fixed steps, which make capacity modeling somewhat challenging. So we're often informed by the clinical team as to the expected arrival of apheresate coming to our facility, which means that we have to have the appropriate resourcing available and the day zero processing team, qualified operators who are ready to attack that. More often than not, there is variance there, either because of the patient's health or scheduling with the clinical site. So we have to be on call for receipt of these materials. The transduction, activation, and expansion of the T cells is more routine, more of a fixed time period. But at the end of the day, there is patient-to-patient variability, which translates into longer periods of time or shorter periods of time. And we have to make sure that we have the resources to support that prior to the harvest and the quality control release and delivery to the clinic. So overall, we have a complex process, a complex technical process, which has both fixed and variable components that makes the challenge of having the resources available challenging. As I think James pointed out, these are individual GMP products, and therefore we have to treat them as individual GMP products in our documentation. I mentioned that there is short notice requests received, process variability, and the challenges in capacity modeling. And this is actually made even more challenging given the fact that we are producing these cells in a qualified aseptic processing environment, which has been qualified with a defined number of operators. And therefore, if we're sloppy in the scheduling, we can have overlap, which would require more staff than can be supported in the manufacturing suite. And that would be not something that we could do in terms of complying with our validation protocols and the EMPQs for the room. So lots of things to think about. Also, just to underline the complexity, these operations take a good while. They use complex equipment. There is the requirement for GMP documentation and verification in batch records. And these are folks who are working for extended periods of time wearing sterile gowning. And it's not unusual for operators to be in the suite standing for 8 hours in their gowns without a bathroom break. So the reality is we have to take these resourcing considerations into mind as we schedule and scale up our capacity. This is a map of a slightly different process that has also a variable unit operation in it. And the way that we are addressing this through the early and later phase 2 clinical studies is to put together workstations in multi-workstation suites and to be able to staff with dedicated staffing to accept these products. And it is reasonable for us to use this model through phase 3, but with some of the indications that we're talking about, where there's tens of thousands of patients that may require dosing long term, there have to be better solutions. And one can look at establishing larger and larger aseptic suites or segregation of expansion activities into gang suites. But at the end of the day, the hopes are that more automated processes and more control over the aseptic environment through barrier technology is going to provide better solutions long term. So we're making Model Ts at this point. We don't have the robots working yet. Obviously, the multi-suite approach does allow for higher throughput without overlap of critical operations. Another challenge is the fact that you have to have the right amount of materials released by your QA unit in order to meet the patient requirements. And when there is uncertainty as to when patients will arrive and what materials are necessary across multiple products, this can be very challenging. Many of the products that we're seeing today have extensive bills of material, 50 or more individual components, biological components, as well as components used in dedicated processing. So maintaining the appropriate levels of these products is a constant headache, and one has to have a very sophisticated system if we're going to be doing this at large scale. And so beyond the facilities, WuXi has invested in supply chain management and MRP systems to be able to support this. This is how we control the supply chain, and it's obviously a very clear linear process. But this is what is required in order to allow materials to be tested, to go through identity testing or extensive testing, activity testing, and to be released for use in manufacturing. Also, biosafety is a challenge, and building the sampling plans to meet both FDA, EMA, KFDA expectations is very challenging. In some cases, surrogate samples are permitted, as in short shelf life biologics or short shelf life cell therapies, where one could take a sample earlier than the final released product, subject it to a rapid testing method, and use that for interim release. With gene-mediated cell therapies involving viral vectors, there is extensive testing required to document the absence of recombinant retrovirus or replication-competent retrovirus or replication-competent lentivirus. And while there's hope that some of the stringency here will be alleviated by FDA, this is the current requirement, and therefore there are extensive facilities required to accomplish this testing. Some of this testing requires cells, co-cultivation assays that will be run for 42 days, 5 passages of cells, seeking to cause the retrovirus to leap out of the cells. So this is another complicated component in our life. With short shelf life products, and many of the products that we're manufacturing today have cryopreservation steps, and we're developing formulations and cryopreservation processes. Some don't. And this is a real-life example of a product that's dispensed at our facility at around noon and is in the patient at 2 o'clock. and the coordination of a GMP interim product release between 2 QA groups and clinical groups is very demanding. We don't necessarily like to do this, but it is possible to have this kind of turnover. Next, I'd like to talk a little bit about what stands in the way of getting there. And some of the folks today have talked about the process of moving to the late stage robust manufacturing at the phase 3 or commercial. That isn't as easy as it sounds. And one of the reasons for that is that there is variability of the source cells. There is limited amount of material which has a clinical correlate available so that processes can be appropriately evaluated or validated, and there are the lack of analytical methods that tie out that allow for comparability. So there are challenges early on presented in moving to a more robust process. There are, as others have pointed out, also limitations on the amount of time the cells can be stored, their stability, and their activity after cryopreservation. In addition to some of the mundane things like, is it a combination product if it's in a syringe, and how do we evaluate the device components associated with that? And lastly, in terms of being able to characterize these products, we find that many do not have robust potency assays. In many cases, there's surrogate potency assays which are being employed. And so all of those present hurdles as we look to move towards developing a robust commercial process. We've accommodated this, and we have had clients who have said, I'm going to stick with my non-robust process until I've gotten approval for the product. And then I will launch the process development activities for the commercial process. We've had other companies which have said, we need to do both an early development PD as well as a late development PD, and we're going to overlap them as much as we can and factor the materials that we need to accommodate that process development effort as we're going. So we've seen both ends of the spectrum. The challenge for us is to have both of those capabilities available to a client simultaneously, which means that our process development staff has grown over the last year by 200%. And then talking about the way that one might partner, these products, by and large, have the regulatory skids greased in one way or another. Clients come to us and say, we're in phase 1 now, or phase 2, but we can very quickly go to commercialization. And that presents a challenge. We need to have available the resources to support a rapid commercial transition. We also have to have capacity, and when one is entering into a commercial manufacturing agreement with a pharma company, there's a little bit of paperwork involved. So these things do add complexity, and the standard approach that's been taken in the past with CMOs of I'll give you some money and you give me a product doesn't work very well. The other challenge that we see and have to address is this compressed window of time for process development and analytical development. And often analytical development is gating our progress in manufacturing. So they have to be attacked simultaneously. And then lastly, our facilities have to be flexible because forecasting launch quantities and market penetration for these products, there are few precedents. So being able to have facilities which are modular and can expand to meet scale up is going to be important. So we've developed strategic manufacturing partnerships where we are able to dedicate suites, we're able to expand in modular fashion as the product demands increase, and able to leverage existing quality control, analytical methods in order to be able to keep this complex process on track. And to some extent, and to much credit to the senior management at WuXi, we have pre-positioned facilities which are built to commercial standards with the faith that many of these products are going to advance rapidly. And we've already signed under agreements manufacturing contracts for products that are in late-stage clinical studies right now. So it's a very exciting time. And while I'm getting lots of gray hair from addressing challenges here and working with our team to address these, we do want to keep in mind, and I'm going to show that slide, show that picture again, why we're doing it. And as James pointed out, this Emily Whitehead, she was the first ALL patient treated at the Children's Hospital in Philadelphia. And she's happily holding a puppy there after 4 years of cancer-free. And there is an Emma Whitehead Foundation if anyone is interested in visiting her website. And lastly, I'd like to thank the folks who are working every day addressing these challenges, putting in long hours to support the active manufacture of these immunotherapy products and the clients' teams that we work with. And with that, I'll say thank you.